Hello and welcome back to Excelsior Academy. So in the previous lecture, we discussed about the cell walls of the bacteria, right? Now we are going to discuss about the eukaryotic cell walls. Now in contrast with bacteria, the cell walls of the eukaryotes, including the fungi, algae, and the higher plants, they all are composed principally of the polysaccharide. So this is going to be very common topic today. So we know these cell walls of fungi, it is made up of N-acetylglucosamine, algae, and the higher plant, the cell walls are made up of cellulose, right? Now, so the basic structural polysaccharide of fungal cell wall is chitin, which also form the shells of the crabs and the exoskeletons of insects and the other arthropods. So this is actually the dried exoskeleton of the insects. So what we do is like for the research purpose, we make it powder of it and then we use it. So I can very well relate with my experience over here. So for my protein purification, so I used chitin. Okay. So chitin was used as for my affinity chromatography. Okay. So it was like, 3 years were over and I was not getting any protein purified. And I don't know, somehow I just thought that maybe the protein is not having any affinity for the single glucose, like single sugar molecules. So I tried with glucose, I tried with fructose, mannose, galactose, जितने भी आपके normal monosaccharide थे मैंने सब try किए थे. Then one fine day, I just thought of why not trying the chitin. So chitin is what? It is your N-acetylglucosamine. N-acetylglucosamine, right? But during the research, uh, it didn't show any affinity towards N-acetylglucosamine also. Okay. But when I was going through all the literature search, okay, I was going on reading. So I came to know that there are certain proteins which might bind to N-acetylglucosamine tetrasaccharide. Did you get it? So yani ki there are 4 residues of N-acetylglucosamine to which my protein was actually able to bind and it had affinity for that. Okay, so जैसे जैसे आपका monosaccharide unit was increasing, okay, it had stronger affinity. So when I did the experiment, I came to know that the protein on which I was working, it had N-acetylglucosamine 4, यानी कि टेट्रासाइक्राइड के ऊपर उसको affinity है, below that it does not have the affinity. Okay, so since they are the polymer of N-acetylglucosamine, the protein was able to bind to the chitin. So then what I did, so we used to buy these shells, okay, and then usko powder banaya, and then I use it for the affinity chromatography. Then all those chromatography steps I carried out, and that's how I was able to purify the protein. So it was very novel, and it was so novel that we didn't have the amino acid sequence of the protein. So we couldn't get the structure then. And not yet any structure is available of that particular protein. आज तक नहीं किसी ने किया है और ना, I mean, of course, even I was not able to do. So the reason which I think this was asked during my PhD viva also, like why people or you are not able to get the structure. So the thing is that this protein is very stable, okay, and it does not precipitate. And for any protein crystal to form, it should be at the border where the protein is either will become a precipitate or it will become a crystal. Okay. But this protein was so stable that it never precipitated. तो वो कभी crystal ही नहीं बना. And without getting the protein crystal, you cannot get the structure also. Okay. So that was the failure during my research. Anyways, now coming back over here. So chitin, it is a linear polymer of N-acetylglucosamine residues. This is the structure. Okay. And glucose. Then you add this amino group, it becomes glucosamine. You add N-acetyl group, it becomes N-acetylglucosamine. Okay. Clear. No need to remember the structure. You can skip. The cell walls of the most algae and higher plants are composed of cellulose, which is the most abundant polymer on earth. Okay, so for if question comes like which is the most abundant polysaccharide, then it is cellulose. Most abundant protein कौन सा होगा? Anyone? Akshaya?
Rubisco.
Correct. Because we have got the higher plants, all over the earth present, and each cell has got the maximum quantity of the enzyme Rubisco, right? So we have got the most abundant protein on earth. Correct, very good. Cellulose is it's a linear polymer of glucose residues, which often contains more than 10,000 glucose monomers, and both in the sugar residues in both chitin as well as cellulose, they are joined by beta-1,4 linkages. Okay, this is important. Please remember. Even if you don't want to remember the structure, you need to remember those. Now coming to the cellulose myofibrils. So then again, see, just a simple leaf, you have got the plant cells, and on the cell wall, you see these. Okay, so these are nothing but the cellulose microfibrils are there. Just like how in muscles we have got the myofibril, but they are arranged, right? Here it is, you can see that the network is present. Now each microfibril is again having these cellulose chains. Okay, you can see it over here. So these chains are nothing but your glucose monomers, which is linked in the beta linkages, beta-1,4 linkages. Okay, if branching is happening, then it is alpha-1,6 linkage. Now, guys, are you able to follow? It's very simple topic today, like only the eukaryotic cell wall. It's simple. I hope you're able to follow, right?
Yes, ma'am. Yes, ma'am.
Okay, thank you. Okay, so within the plant cell wall, cellulose microfibrils are embedded in a matrix consisting of proteins and the two other types of polysaccharides called hemicellulose and pectins. Now, this is something which you have to understand, okay? So hemicellulose kya ho gaya? Again, to the glucose units, you have got the other monosaccharides coming and binding, okay? Here, xylose, galactose, fucose, all these sugars are present. That's why it is hemicellulose. Now, coming to the pectin, again, in the case of the pectin, you have got galacturonic acid. This stands for galacturonic acid. And then we have got rhamose. Okay. Now, what you see is that the pectin has got negative charge. Okay. And among the cellulose का ऐसे जो network रहता है, in this you have got between the hemicellulose present. And we have pectin. Okay, like this present. So this pectin is having the negative charge. So in order to stabilize the entire structure, the pectin interacts with your calcium 2 plus. Okay. Now because of this negative and positive interactions, they retained water and result in a gel-like structure. Okay. Now let's go slowly again about this. So the hemicelluloses are highly branched polysaccharides that are hydrogen bonded to cellulose microfibrils. This stabilizes the cellulose microfibrils into a tough fiber, which is responsible for the mechanical strength of the plant cell wall. Okay. Now, cellulose microfibrils and hemicelluloses are embedded in a gel-like matrix formed by the pectins. Now, these pectins, which are branched polysaccharides, contain negative charge because of the galacturonic acid. ठीक है, हमने यहाँ पे acid group present है. If you see the structure here, this is your galacturonic acid where you have got COO minus group is there. That's why it is negatively charged. Now, because of these multiple negative charges, pectins bind positively charged ions such as calcium and trap water molecules to form the gel. So that's why whenever you, if you try to tear the leaf, okay, you see that, that mucus jaisa ya that chip karne wala sa aise gummy type things comes out. That is because of the pectins. Okay. Now this is how they are arranged. So in the plant cell wall, you have got the middle lamella, primary cell wall, and the plasma membrane, right? Now see, These bigger things are your cellulose microfibrils. This one. Okay. Then in between you have got these ones, sorry, these green color things. This are the hemicellulose. So as just now we saw that hemicellulose makes hydrogen bonds with the cellulose microfibril, right? Then we have got the pectins. So here pectins are also present. Now because of this all arrangement, now pectin negative charge associated with calcium 2 plus will trap water molecules. Okay, so this entire region is present in a gel-like component. Okay, and then certain other soluble proteins are there. Clear? Okay. Now let's talk about the turgor pressure. So whenever anywhere we discuss about the cell wall of a plant cell, you need to talk about the turgor pressure, because this is very important in maintaining the shape of the plant cell, right? And it is also responsible for, the growth of the plant and the movement of the water. So turgor pressure. So turgor pressure, it is the pressure exerted by the fluid in a cell that presses the cell membrane against the cell wall. Okay, so it is also responsible for much of the rigidity of the plant tissue. It provides basis for a form of cell growth which is unique to plants. So इसका क्या मतलब हो रहा है? It is saying that see क्या होता है turgor pressure में, if suppose this is your cell wall, okay, you have got the nucleus, and in addition to this, in the case of plant, you do have the vacuole, right? Now, kaisa hota hai? If you keep on adding, if you keep on taking water over here, what will happen? The size of the vacuole is going to expand, right? Now, this puts a pressure on the cell wall to increase. ठीक है? तो cell wall आपका expand होता है. Now, this is going to expand. It means what? The cell size is increasing. Eventually, the entire tissue is increasing, and the organ or the leaf or whatever, whichever part of the plant, it is going to increase. So that's why it is also providing the basis for a form of cell growth. And this can only happen in the plants. Okay. Now, in particular, the plant cells frequently expand by taking water without synthesizing new cytoplasmic components. Same thing we just now explained. Yeah, coming over here. So same thing, expansion of the plant cells. So because the turgor pressure, what will happen? The vacuoles are there. They will start taking the water molecules and they will tend to expand. Okay. So now this is totally getting accumulated inside the vacuole and they are expanding, because of which the cell wall is also expanding. Now the question is here, when the cell wall is expanding, okay, does it mean that just now we said that the cell without the synthesizing any cell components, did you see? See, the plant, oh, sorry. The plant cells frequently expand by taking a water without synthesizing new cytoplasmic components. So what exactly then, not what exactly, sorry, how exactly then the cell wall is expanding? Any guesses? So right now what we said, see in the case of animal cells, अगर उसको expand करना है तो हम कैसे करते हैं? That the ER and the Golgi are responsible in synthesizing the phospholipids, which are then transported to the plasma membrane. They double in size and that's how they divide, okay, or they grow. Now, when we are saying that there is no cytoplasmic synthesis happening, then how come the cell wall is also expanding when the vacuole is expanding with the uptake of water? Is the question clear to everyone?
Yes, ma'am.
Okay. Now think about it. क्या reason होगा कि without synthesizing, how the cell wall from this much small, it has gone to this much big without synthesizing any cell wall components?
Ma'am, maybe cell wall is stretchable, and when the water they are, when the water, they are taking up the water, so they are becoming thin, and then
Absolutely correct. Very good. So here what is happening is the rigidity of your plant cell wall is because of the the rigidity of the plant cell is because of the cell wall, right? Now, as she said, the cell wall is actually getting loosened up here. जो इतना मोटा cellulose microfibril था, now that is getting thin. It is getting stretched. Okay. So absolutely correct. Yeah. So that's how the cell wall is actually getting stretched or expanded. So cell expansion by this mechanism, it's signaled by plant hormones, especially the auxins, which activate proteins called expansins. Okay. So they are going to help in the expansion of the cell wall. So the expansins, they act to weaken a region of the cell wall, allowing turgor pressure to derive the expansion of the cell in that direction. ठीक है? अगर आपका vacuole ऐसे बढ़ रहा है, then obviously your cell wall should also increase like this, not like this, right? Then it is not going to make any sense. As this occurs, the water that flows into the cell accumulates within a large central vacuole. So the cell expands without increasing the volume of its cytosol. So such expansion can result in 10-fold to 100-fold increase in the size of the plant cells during the development. Good. So this expansion is done with the help of the proteins called expansins. Now look at over here. If these two are the cellulose microfibril, and then you have got many attachment. Okay. Now what is happening is that when this expansion protein is coming, I mean it's blurred, the image is blurred. What they are doing is the distance between the two is being increased. See, they remove this interactions because of which the distance is increased here, right? So this can be done by the expansion protein. I guess I have another image. Yeah. So you can see it over here. We have taken this small section over here, and then let's see, in the case of the normal epidermal cell wall, what is happening? What is happening? You have got over here the membrane, everything present, right? And these are your cellulose and the other portions, hemicellulose, pectins, etc. You can see that how nicely they are compact, right? It's a compact structure. But once you have got the expanding epidermal cell wall, then it gets loosened up with the help of these expansion proteins. Okay, so they make sure that the distance between the two microfibrils is increased, and that's how they loosen up, and it is easy. जब कोई चीज लूज रहती है उसको stretch करना आसान होता है, right? And that's how it is getting the increase in the cell wall. Okay, good. Okay, now as cells expand, new components of the cell wall are deposited outside the plasma membrane. अब जैसे जैसे ये expand होता जाएगा, now let us say that the cell wall, now the cell wants to synthesize, ठीक है? अब हम क्या करेंगे? We want the cell wall components. Now these cell wall components, including hemicellulose, pectins, all are synthesized by the Golgi apparatus. ठीक है? Cellulose is synthesized by the plasma membrane enzyme called cellulose synthase. Please remember, this is really very important. So cellulose is by the plasma membrane enzyme. अब देखो ये आपका plasma membrane हो गया, बाहर आपका cell wall हो गया, correct? So this enzyme is present here. Cellulose synthase is present here. It's a transmembrane enzyme. ओके तो ये यहाँ से सिंथेसाइज करके यहाँ से आपको यूनिट देता रहेगा सो दैट इन दी सेल वॉल इट कैन बी एडेड. Cellulose synthase is a transmembrane enzyme. It synthesizes cellulose from UDP glucose in the cytosol. The growing cellulose chain remains bound to the enzyme as it is synthesized and translocated across the plasma membrane to the exterior of the cell through a pore created by multiple enzyme subunits. So what we are saying here that आपने यहाँ पे UDP glucose से बनाया cellulose components, then there's a pore, it goes out and then gets inserted into the cell wall. Okay, now look at over here. So you have got here the cellulose synthase. Look at the structure. So this is your cellulose synthase. Now what is happening? then the UDP glucose is coming as your substrate. Okay, it is getting synthesized and it is forming your cellulose microfibril together. Can you see here, here, here? All your cellulose synthase is present in the plasma membrane. So once they are formed, it goes and it becomes a part of your cell wall. Okay, is this much is clear? Okay, yeah, able to follow?
Yes.
Okay, okay, good. Okay, there's this animation. Let's watch this also. Now it is visible, the screen.
Yes, ma'am.
Okay.
The cell walls of most algae and higher plants are composed principally of cellulose, which is the most abundant polymer on Earth. Within the plant cell wall, cellulose microfibrils are embedded in a matrix consisting of proteins and two other types of polysaccharides. Cellulose microfibrils are bundles of cellulose chains. A cellulose chain is a linear polymer of glucose residues, often containing more than 10,000 glucose monomers. Both the structure and function of cell walls change as plant cells develop. The walls of growing plant cells are called primary cell walls and are relatively thin and flexible, allowing the cell to expand in size. In this example, the cellulose fibers of the primary wall appear to be randomly arranged, a feature that would allow the plant cell to expand uniformly in all directions. Once cells have ceased growth, they frequently lay down secondary cell walls between the plasma membrane and the primary cell wall. Such secondary cell walls frequently consist of 3 layers and are both thicker and more rigid than primary walls, a feature particularly important in cell types responsible for conducting water and providing mechanical strength to the plant. The cellulose fibers of secondary walls are highly ordered. The cellulose fibers of each layer differ in orientation, forming a laminated structure that greatly increases cell wall strength. During cell growth, plant cells frequently expand by taking up water into a large central vacuole without synthesizing new cytoplasmic components. Cell expansion by this mechanism is signaled by plant hormones that activate enzymes to weaken the cell wall. As cells expand, new components of the cell wall are deposited outside the plasma membrane. A complex in the plasma membrane called cellulose synthase creates new cellulose chains. Cellulose synthase is a transmembrane enzyme that synthesizes cellulose from UDP glucose in the cytosol. The growing cellulose chains remain bound to the cellulose synthase complex as they are synthesized and translocated across the plasma membrane through pores in the multiple enzyme subunits. In expanding cells, the newly synthesized cellulose microfibrils are deposited at right angles to the direction of cell elongation, an orientation that is thought to play an important role in determining the direction of further cell expansion. Interestingly, the cellulose microfibrils in elongating cell walls are laid down in parallel to cortical microtubules underlying the plasma membrane. In these elongating plant cells, microtubules have been stained so that they have a red fluorescence. In the same cells, the cellulose synthase enzymes have been tagged so that they fluoresce green. Images taken over a 30-second interval show cellulose synthase tracking along the microtubules. A merged image illustrates the co-localization of cellulose synthase and microtubules. By guiding the orientation of newly synthesized cellulose microfibrils, cortical microtubules define the direction of cell wall growth, which in turn determines the direction of cell expansion and ultimately the shape of the entire plant.
Okay. Yeah. Clear? So guys, is it clear to you? Okay, so the next topic is about the extracellular matrix and the cell matrix interactions. So here we stop with the eukaryotic cell walls. So as I said, this topic, this unit is about the cell walls, extracellular matrix and the cell-cell interactions. So now we're going to discuss the second topic, that is extracellular matrix and the cell matrix interactions. Okay, so although animal cells are not surrounded by cell walls, most animal cells and tissues are embedded in the extracellular matrix. And this matrix, they help in binding the cells together as well as the tissues together. Now look it over here. So this is your plasma membrane. We have talked about the microfilaments and integrin molecules connections, right? Now they are in turn are connected to the extracellular matrix components called like proteoglycan complex or the collagen fiber, right? So all these components are present in the extracellular matrix and their connections we will be discussing in the further. Now, the first component is your matrix structural proteins. One second, guys. Okay. So the matrix structural proteins. So the major structure protein of the extracellular matrix is collagen. Now, this is really very important, which is the single most dependent protein in the animal tissue. Okay, so there's one more thing, that when the cell or the extracellular matrix nearby of that cell loses collagen, okay, then it is difficult to replace. If it is a loss of the cell, then na cell to divide hoke wapas aa jayega. But if this extracellular matrix is lost, then it is gone case. So you might seen that jab kabhi suppose there is a scar, okay, koi wound ki wajah se scar ho gaya hai, so what we see is that almost this much portion is gone, right? So it never comes back because the cells as well as the extracellular matrix present over there, everything is gone, right? So it will just heal up, but that gap is not filled. Jaise plant mein kya hota hai, if you make a cut, that particular region will come back. If you take out one leaf, right, suppose यहाँ से एक leaf आपने ले लिया, so after some time another leaf will come, but in the case of animal tissue it is not possible, right? You know, now you might have seen that in the faces, like human faces also, many people are there, like because they face so much of acne problem, like if you know any person like that. So because of the continuous acne problem, what happens is that, you know, there is a loss of those particular cells and the extracellular matrix present there, and eventually results in the gaps like craters and all. So that is because of the loss of the collagen over that area. Now what is the treatment? Either you go for the peeling stuff and go for the injections. So especially if you have got the collagen present, your face looks very brighten up, okay, it looks good, shining, and with the age you start losing all this thing, and that's why the all wrinkles and all starts. Okay, so you might have known like many people are there who takes injections, right? So those are nothing but they are the Botox injections. What they take is they actually fill the gaps between this extracellular matrix so that the same fullness is seen again, it's regained since there is a loss of collagen. Okay, coming back to the theory. So they are characterized by the formation of the triple helices in which 3 polypeptide chains are wound tightly around one another in a rope-like structure. So there are 3. So ऐसे आपके 1, 2 and 3. Okay. So 1, 2, 3 alpha helices are present in the collagen. Now the triple helix domain of collagen, it consists of the repeats of the amino acid sequence called glycine XY. This is important, guys, okay? In collagen, this domain is important. So one is glycine. So it is the smallest amino acid, as you know. Now at every third position, glycine XY, again यहाँ पे glycine आएगा. ठीक है? Now we need to see what X and Y can be. So proline is frequently found in the X position and hydroxyproline is found in the Y position. Now, because of the ring structure, these amino acids stabilizes the helical conformation of the polypeptide chain. So it's highly coiled structure, okay, highly coiled structure it is. Now, because of the ring structure. So, जब भी आप देखोगे alpha helices में जब भी जहाँ पे at the moment it is making a turn, okay, for your alpha helical formation, it is always because of the proline. Okay, so now we know in the case of the collagen, it is your glycine, proline, and hydroxyproline. Okay, now the unusual amino acid hydroxyproline is found within the ER by modification of proline residues that have already been incorporated in collagen by polypeptide chain. So what does it mean? It means पहले आपका collagen बनता है जिसमें आपका glycine रहता है, proline रहता है, and proline रहता है. Later this gets modified. Okay, now there are a few cases lysine residues are also present which later gets converted to hydroxyl lysines. These are the modified amino acids found in the collagen. The hydroxyl group of these modified amino acids are thought to stabilize the collagen triple helix by forming hydrogen bonds between the polypeptide chain. So as I said, if suppose you have got like this, so even these are in making interactions. So these interactions are possible because of these hydroxyl group, OH group. Okay. Now, so this is how your collagen triple helix looks like. So 1, 2, and 3. And if you see the sequence, glycine, proline, serine, glycine, proline, arginine, glycine, proline, hydroxyproline. Okay. So it wasn't means that glycine XY बोला था, right? So glycine तो आपका present है ही at the third position. This is by default it is present. X and Y can vary, but most of the time what we have seen is that X is proline and Y is your hydroxyproline. Okay, but we can have other amino acids also present. Is it clear, everyone? Tanvi? Akshaya?
Yes, ma'am.
Okay, this is something which you have to remember. Yehi question mein aayega. Now, this is how the collagen fibrils looks like. Okay, so here this is triple helical structure of collagen. Then again, they are cross-linked with the another one which is present over here. Okay, then this is the again the electromicrograph of the collagen fibrils. And thus, so they are arranged. You can see it over here, like they make the network also. Okay, now there are 5 common types of the collagen. Type 1, which is present in the bone and skin. Type 2 is present in the cartilage. 3 present in the connective tissue. 4 in the basement membrane. And the 5th one in the hair. Okay, this is something easy to remember. There are 5 types are there present in the bone, skin, cartilage, connective tissue, basement membrane, and the hair. So 5 types of collagen is there. We don't have to go in the details, so that's okay. Okay. Sorry. Now, connective tissue also contain elastic fibers. Now, this elastic fibers, they are very important. नाम से पता चलता है elasticity है. So यानी कि ऊपर नीचे, they can stretch it, they can come back to its original shape, then again stretch it, come back to original shape, right? So that is their function. For example, in the case of the heart, it always beats, right? So it is again stretching and coming back, stretching and coming back. Lungs, you exhale and inhale. What is happening? You're expanding your lungs, then you're relaxing. Expanding your lungs, then relaxing. So there you have got extensive network of the elastic fibers. So connective tissue also contain elastic fibers, which are particularly abundant in the organs that regularly stretch and return to their original shape. For example, the lungs stretch each time a breath is inhaled and return to their original shape with each exhalation. Now, elastic fibers are composed of protein called elastin. Now, this is again cross-linked into a network by covalent bonds found between the side chains of the lysine residues. This is something which you have to remember. So, elastic fibers made of elastin and they are cross-linked at the lysine residue. Okay, so they behave like a rubber band stretching under the tension and then snapping back when the tension is released. Now that was all about the protein, matrix proteins. Now coming to the matrix polysaccharides. Okay, so it's a fibrous structure proteins of the extracellular matrix embedded in the gels, which is formed by the matrix polysaccharides called glycosaminoglycans, which consist of repeating units of the disaccharides. So they are your GAGs, glycosaminoglycans. Okay, now one of the sugar is either N-acetylglucosamine or N-acetyl-galactosamine, and the second is usually a acidic glucose, either glucuronic acid or hydronic acid. Okay, ऐसे आपको याद रखना है N-acetylglucosamine or N-acetyl-galactosamine. Okay, plus any acid. That is either glucuronic acid or hydronic acid. Now there are certain exceptions are there in which, for example, with the exception of hyaluronan, these sugars are modified by the additions of the sulfate groups. So because of the sulfate groups, what happens? Then they become negative charge. Now, as we saw in the case of the plant cell, the pectins were having glucuronic acid, right? So they were also having the negative charge. This negative charge was in association with calcium 2 plus retaining the water, and hence it was becoming a gel-like structure, right? Same way, these negatively charged GAGs, they also have negative charge. So they bind positively charged ions, again trapping the water molecules to form the gel. Okay, and hence they provide the support to the extracellular matrix. See here. So we have got the other glycosaminoglycans which is present in your extracellular matrix. Okay, so disaccharide units. Hyaluronan, in which glucuronic acid and N-acetylglucosamine. Dermatan sulfate, it is hydronic acid and N-acetyl-galactosamine. If it is conductan sulfate, glucuronic acid and N-acetylgalactosamine. It's just the combination of these four only. Then galactose, N-acetylglucosamine for the keratin sulfate, heparan sulfate, hydronic acid and N-acetylglucosamine. Yes, and the question whether you have to remember all this, yes, you have to remember not the structure but the composition, like dermatan sulfate में क्या present है, keratin sulfate में क्या present है. Okay, so all this you have to remember. This is important, very, very important. Okay. Now, where they are present in the extracellular matrix. So the common sulfated GAGs are the dermatan sulfate, conductan sulfate, keratin, and the heparan sulfate. Hyaluronan is the only GAG that occurs as a single long polysaccharide chain. Rest all are the disaccharide. Okay. So like cellulose and chitin, it is also synthesized at the plasma membrane by a transmembrane hyaluronan synthase, just like your cellulose. So this is a proteoglycan. As you can see, you have got the core protein here, and to that your GAG chain, that is glycosaminoglycans, start binding. Okay, they start forming the linking. So this can be either conductan sulfate or it can be your keratin sulfate. So that's how all these matrix polysaccharides are present in the extracellular matrix. Okay, did you get it? So having the core protein and to which the GAGs are joined, those are known as the proteoglycan. Yeah, that's all guys for today. I'll be stopping here now.